following video will cover subtopic 2.4, proteins. Here is a list of understandings, applications, and skills that we will cover in this video. So we'll start with the general backbone of a protein. So proteins are made up of amino acids. So this diagram here just goes through the general structure of an amino acid. So just recall we have that central carbon that is bonded to 4 different things. So it is attached to a carboxyl group. On the other side, it is bonded to an amino group. That central carbon is bonded to a hydrogen. And then the 4th thing is the R group, which will specify the type of amino acid that we are dealing with. And so amino acids will link together. So you'll have the carboxyl group joining together with the amino group to create what is called a peptide bond. So in that carboxyl group, you'll have an OH group leaving, and from the amino group on the adjacent amino acid, you'll have a hydrogen leaving together. They will form water. The fact that we have water being formed, this reaction is referred to as a condensation reaction. And then the whole thing here is the peptide bond itself. So the carbon double bonded to oxygen, and that carbon single bonded to nitrogen is the peptide bond. And so you can have a whole string of amino acids bonded together through that peptide bond, which will produce a polypeptide chain. So a polypeptide chain is just many amino acids linked together. So, poly meaning many. And then the peptide is going to be made of our amino acids. So this animation here just goes through the formation of a dipeptide through that condensation reaction. So here we have water being created, and then the remaining part of the amino acid joins together, which is now the peptide bond. So one of the things you'll be expected to do is to be able to draw a molecular diagram to show the formation of a peptide bond. So we'll just go through the steps here that you can use to create this drawing. So there is a chain of atoms that's going to be linked together by a single covalent bond, forming a backbone with a repeating sequence of NCC. So you need to look for that NCC or create that NCC sequence to start with to create that backbone when creating that peptide bond. You have a hydrogen atom that is linked by a single bond to each nitrogen in the backbone, and an oxygen atom linked by a double bond to one of the carbon atoms. The amine and carboxyl groups are used in forming that peptide bond. So in this example here, we'll draw a peptide bond between alanine and serine. And so if we look at alanine here, we can identify the different parts of our amino acid. So here we have the carboxyl group. Here we have the amino group, and then this CH3 group is our R group. In serine, we can do the same. We can identify our carboxyl group. We can identify our amino group. Obviously, we have our hydrogen. And then the CH2OH is going to be the R group. So when we draw the formation of that peptide bond, you want to draw that NCC backbone chain of atoms. So in each amino acid, there is this chain of atoms, the N from the amino group, the central carbon, and the carbon from the carboxyl group. Here's our NCC. And then when we draw our next amino acid, we just repeat that sequence of nitrogen, carbon, carbon. So we can draw nitrogen, carbon, carbon. And then we can fill in the rest as well. So on our terminal amino group, on our end amino group, we can draw it with two hydrogens attached to the nitrogen. And then we have our central carbon. So off of that next carbon, we have our hydrogen, and then our first R group. And then we have on the next carbon, that is double bonded to oxygen. And then we repeat for our next amino acid. So that next nitrogen, though, is only going to have one hydrogen bonded to it. And then the next carbon is going to be that central carbon for serine. That's going to be bonded to hydrogen. And then that will be bonded to our R group. And then the last carbon is going to be our carboxyl group. So here we have our peptide bond. And then we should also show the other product of this reaction, this condensation reaction, which is water. So here, the hydroxyl group, this OH group from the carboxylic acid, is going to be removed. And a hydrogen from this amino group is going to be removed, and these two will combine to form the water. So the formation of a water is that condensation reaction. So there are 20 different amino acids in total, and these 20 different amino acids can be used in any sequence to build polypeptides. And those polypeptides, those amino acids, are assembled together in those polypeptides on ribosomes. So here are the 20 different amino acids. The highlighted parts for each of them are the R groups that specifies the type of amino acid that we are dealing with, or the specific amino acid that we are dealing with. And keep in mind that you do not memorize these R groups for the amino acids. So you need to be able to draw the general structure, and that is it. What you do need to know is that you have amino acids that exist as nonpolar amino acids, and we'll group these guys together as polar amino acids. So you have nonpolar, that's off to the side, and I'll rewrite it. So you have nonpolar amino acids, and you have polar amino acids. And you need to be able to explain how these amino acids would make up a protein structure. So, for example, where you would find these different types of amino acids is in an integral protein. So hopefully you recall what an integral protein is from the first unit. So it is that protein that is going to span that cell membrane. In this case, the integral protein that I'm going to make is a protein channel. So we'll draw those two parts to it. Finish off the membrane here. So here is our protein channel. Here's the channel itself. So just remember that proteins, here's a protein, are made up of amino acids. So all these amino acids here are combined together in different orders to create proteins. And so the fact that we have polar and non-polar amino acids means that we can predict where we will find certain types of amino acids. So recall that when we talk about a non-polar amino acid, this is also going to be a molecule that is hydrophobic. or water-fearing, and our polar amino acids are going to be hydrophilic or water-loving. So in a protein channel, you can imagine that these portions, the portions of the protein channel, so I'll just outline this in purple here. These portions of the protein channel, we would expect to contain polar amino acids, because these are exposed to the aqueous environment and this extracellular and intracellular environment. And remember that in that extracellular environment, there is a lot of water, and even inside the cell, there is going to be a lot of water. So the amino acids that you want in contact with that aqueous environment, that environment containing water, would be water-loving amino acids. So you'd find more polar amino acids on the outside of that protein channel in contact with water. On the areas of the protein that would be embedded in the membrane, and I'll use red, these portions here that are in contact with this hydrophobic core, for that protein to preferentially interact with that hydrophobic core, you would want nonpolar amino acids in there. So in this part of the protein channel, you're more likely to find nonpolar amino acids. And then we'll talk about it later in the video, but this protein channel is going to be a three-dimensional structure, so there's going to be amino acids even on the inside of this protein channel, and those amino acids will tend to be non-polar amino acids as well. But using polar and non-polar amino acids, categorizing them in that manner, we can predict where they will be found within a protein. And so you do not need to know which amino acids are polar or non-polar, but just simply that amino acids can be polar or non-polar. So now, with those proteins that get created, remember it's ribosomes. The main job of a ribosome is involved in protein synthesis. So how does that ribosome synthesize those proteins? Well, a ribosome will link those amino acids together. So ribosomes are the molecules within cells that facilitate the formation of a peptide bond. Remember, a peptide bond is what's going to hold those amino acids together. So a ribosome will bring together amino acids. Here are amino acids, and it will facilitate or create a peptide bond between these amino acids. And so that is the main job of what that ribosome will do. Now, with these polypeptides, as they're being built, the amino acids that are used to build them, they can be linked together in any sequence, which gives a huge range of possible polypeptides. Remember, there are 20 amino acids, and we can link them together in any sequence we want. We can repeat as many amino acids as we want. So it gives a very, very large range of possible polypeptides or proteins. And this is also due to the fact that peptide bonds can form between any pair of amino acids. And then the number of amino acids in a polypeptide can be anything between 4 and tens of thousands of amino acids. I think the most common number is somewhere in the hundreds to low thousands of amino acids. But the fact that you have such a huge range, and it can be any sequence of amino acids here, this gives almost an infinite number of possible unique amino acid sequences. And so the sequence of amino acids is going to be important for that particular polypeptide. Um, a particular polypeptide or a particular protein will have a unique amino acid sequence. The code or the instructions for that amino acid sequence is coded for in DNA, and it is coded for by genes that are in DNA. So a gene is a unit of inheritance consisting of a specific sequence of DNA. So there is a specific sequence of DNA. For every protein, there is a specific sequence of DNA that will provide the instructions for what sequence of amino acids is needed. And so information for that amino acid sequence of a polypeptide is stored in that sequence of DNA. This leads us into that central dogma of genetics. So basically, anything that makes all living things are going to have DNA as where it stores its genetic information. So within the DNA is where we find those genes which code for proteins or polypeptides. DNA does not get used directly in those instructions. Instead, a message molecule is made called RNA. This carries the message of the gene and gets used by the ribosome to synthesize the polypeptide or that protein, and this is the end product. And so there are several events that have to occur in order to get that protein created, which we will go through later on in this unit. But then keep in mind that within a cell, we're talking about a eukaryotic cell here, that the DNA is going to be stored within the nucleus. The message gets created within the nucleus. That message molecule is exported to the cytoplasm, and it's in the cytoplasm that the ribosomes, this circle here is a ribosome. It's in the cytoplasm that ribosomes will use the message to create a protein. So protein synthesis will happen in the cytoplasm. Now, within a protein, there are levels of protein structure. So there's different categories of protein structure, or 4 levels of protein structure. So the amino acid sequence of that protein is going to determine the 3-dimensional structure of the protein, or the 3-dimensional conformation of that protein. So keep in mind that proteins, when in proper conditions, are going to be stable, folded 3-dimensional structures of these polypeptides. Now, there are 4 levels of protein structure. There is the primary structure, secondary structure, tertiary structure, and quaternary structure. All polypeptides have at least up to the tertiary, and then certain polypeptides or certain proteins will have that quaternary structure. So with the primary structure simply refers to the order and number of amino acids in a polypeptide chain. So it is the sequence of those amino acids in that polypeptide chain. So the primary structure is read from the amino terminal to the carboxyl terminal. So you read it from the NH2 group all the way to that end carboxyl group. And then each amino acid is going to be identified by its specific R group. And then most polypeptides are between 50 to thousands of amino acids long. So the primary structure is basically, each amino acid within that sequence that is linked in there. The secondary structure, this one's a bit more complex to visualize, but it's referred to as the local folding within the polypeptide backbone. So that NCC sequence is going to fold in particular ways, which will be stabilized by hydrogen bonds. So there are 3 types of secondary structures. There's what's called the alpha helix, So it forms this helical structure. So here you can see the CCN, the NCC, NCC, which forms this helical structure, which are stabilized by hydrogen bonds. There's beta pleated sheets where basically like the backbone forms this kind of accordion shape to it, and will interact with another part of the backbone at a different portion or a different location in the primary structure. So it folds this pleated sheets. And then there's what's referred to as open loops, which are literally just loopy sequences of amino acids. And so the next level of protein structure is the tertiary structure. And the tertiary structure is the three-dimensional conformation of a polypeptide. So this is where the amino acid chain links itself in place in the form of unique twisted or folded shape of the protein. So this is where all those local structures will kind of take a particular position in space, and it will give the protein a particular shape. So in this example here with myoglobin, this is the tertiary structure of myoglobin. So you can see the secondary structures here, these alpha helices, that are oriented in a particular direction in space. And this particular orientation will give myoglobin its unique shape. The tertiary structure is also going to be stabilized by R group interactions. So those R groups for specific amino acids are going to interact together to help stabilize this tertiary structure. And in the tertiary structure is where we tend to find a hydrophobic core. So, somewhere in the middle of this protein here, we'll have a higher tendency of finding non-polar amino acids. And the 3D structure is going to be important because it is going to give the protein its particular functional properties. So whatever it is that protein is going to do, it needs that three-dimensional structure in order to give it its functionality. So when we talk about enzymes, enzymes which speed up reactions, they have a particular shape to them. The shape of that enzyme is going to be important for it to do its job. The last level of protein structure is the quaternary structure. And the quaternary structure is basically a number of tertiary polypeptides joined together. So you take a number of individual polypeptides that are often like the same or very similar, and they are linked together or interact together to form that quaternary structure. So hemoglobin is an example of a quaternary structure. So hemoglobin is what carries oxygen within red blood cells. Hemoglobin is composed of 4 different polypeptide chains. Each chain forms a tertiary structure called a heme group, and all 4 of those heme groups together is what creates hemoglobin. Hemoglobin also has what is called a prosthetic group, which are proteins that are bound to inorganic groups. So it's kind of like little accessories. So hemoglobin has 4 polypeptide heme groups, and these heme groups are each associated with an iron ion at the center of it. And the iron ion, iron 2 plus ion, attached to that heme group is where oxygen is going to interact with this molecule. So the way to kind of think about quaternary structures, because it might be hard to visualize, is if your transformers, they're the constructicons, and then there's the individual constructicons here. So these guys here would be like the individual polypeptides, these individual like tertiary polypeptides here, and then they can function. They can somewhat function individually, but ultimately, like, they need each other to come together to function as one unit, and this is where they function most efficiently here. So, all of these constructicons, they can merge together to form Devastator. which is sort of like the quaternary group here. And then, Devastator has his like little cannon here, which he can use, and this is kind of like his version of a prosthetic group. It's his accessory. If you're not familiar with Transformers, maybe you're familiar with Mighty Morphin Power Rangers. So like, same thing here. Each of these Zords, would be the individual peptides, and those Zords come together to form a bigger structure. Megazord, which is that quaternary structure, and then Megazord has his like sword and elephant head shield, or I guess mammoth shield, which would be like the prosthetic groups. Those are those accessories that help him do his job. And so with these protein shapes now, so these tertiary structures and these protein shapes can often be described as being fibrous or globular. So fibrous proteins will have a structural role, whereas globular proteins are more functional. And so this chart here just goes through fibrous proteins and globular proteins and various properties of them. So fibrous proteins tend to be long and narrow. They have that structural role in strength and support. And because they provide strength and support, they tend to be insoluble in water. They need to be able to hold things up, so we don't want them dissolved in water. And so a fibrous protein will have a repetitive amino acid sequence, and they tend to be less sensitive to changes in heat, pH, and other conditions. And examples of these fibrous proteins include things like collagen, which are found in skin tissue, myosin, fibrin, actin, which is found in muscle, keratin, and elastin. Globular proteins, instead of long and narrow, tend to be rounded or spherical in shape. They have more of a functional role, so like they tend to speed up reactions, a catalytic role, or involved in transport, so transporting materials in and out of a cell, so protein channels, for example, and as a result, they are generally soluble in water. They have an irregular amino acid sequence, so their amino acid sequence is not repetitive. It just seems kind of random from beginning to end. And as a result, they are more sensitive to changes in heat and pH. And then common examples include the enzyme catalase, hemoglobin, which we discussed on the previous slide, insulin, which is involved with glucose, blood glucose regulation, and immunoglobulins, which are antibodies. Antibodies we've talked about previously, but antibodies have a function in the immune system. And so if a protein has a particular shape and has to function with a particular shape, that also means the protein can lose its shape. And when a protein loses its shape, this is referred to as protein denaturation. And so denaturation is that process in which the protein unravels and will lose its native shape, its original intended shape. When a protein denatures, when it unravels, the protein will now become inactive. And this is, and in that process of denaturation, why that happens or why it will unravel is that the interactions between R groups get interrupted. And so there are these different R group interactions that will help hold that protein in place for its tertiary structure. So here is our protein. This is our purple line. The purple line itself would just be the primary structure. And then the specific shape that this primary structure takes is going to be the tertiary structure. And the tertiary structure is going to be held in place by these various R group interactions. So R groups that are capable of forming hydrogen bonds are going to help stabilize the structure. In these areas of the protein where you find nonpolar amino acids, the R groups on the nonpolar amino acids are going to tend to interact preferentially with each other. So these are the hydrophobic interactions. amino acids that have positive and negative charges on them in their R groups are going to associate together to create ionic bonds between different portions of the protein or different portions of that amino acid sequence. And then you can have what are called disulfide bridges. These are amino acids that have sulfur at the end of them here. And those sulfurs will bond together, create a covalent bond together. And so you'll notice that these different R group interactions, like these amino acids, are located at different spots in the primary sequence. And so the folding over of the chain itself allows these amino acids to interact together. By disturbing these R group interactions, this can cause the protein now to unfold and unravel so that these R groups are no longer interacting with each other. And so something like pH can affect these R group interactions. So at a high pH, where you increase hydrogen ion, or sorry, at a low pH, where you have high hydrogen ion concentration, these hydrogen ions can come in here and disturb that interaction between these hydrogen bonds. It can mess up the ionic bonds themselves, and this can disrupt these R group interactions, causing that protein to unfold. And so proteins carry out a number of different functions within a cell, depending on the cell that it's expressed in. So, they can be involved in catalysis, speeding up reactions. They're involved in muscle contractions. They're involved with the cytoskeleton, cell adhesion, membrane, transport through protein channels and protein pumps. Involved with immunity, for example, with antibodies. Involved in tensile strength, blood clotting, transport of nutrients and gases. So, for example, transport of oxygen or CO2. That's what hemoglobin does. Receptors, hormones, and packing of DNA. Those are histones. So, a number of a variety of things that proteins do within a cell. And so there are 6 specific proteins that Ivy wants you to know about. So that's ribisco, insulin, immunoglobulins, rhodopsin, collagen, and spider silk. So you need to know what they do. However, you do not need to know the structure. So the detailed structure of these proteins is not needed. You just need to know what they do, what their function is, why they're important. And so information on this can be found on page 94 of the textbook. Please read about them, make notes, and memorize. In the nature of science part of this subtopic, this is looking for patterns, trends, and discrepancies. So most, but not all amino acids assemble proteins from the same amino acids. So some organisms will use amino acids outside of those common amino acids, and this can include amino acids such as selenocysteine or pyrrolysine. More information on this can be found on page 89. So here is the common amino acid cysteine. So cysteine has sulfur as part of the R group. So here is the central carbon. Here is the amino group. Here is the carboxyl group. This part here is the carboxyl group. And then this portion here on cysteine is the R group. At the end of this R group is a sulfur atom. This is what gives cysteine its ability to form disulfide bridges. And we can get the uncommon amino acid selenocysteine, which has the same structure, but instead of sulfur, it now has the atom selenium attached at the end instead of sulfur. And then you can have the amino acid pyrrolysine. So lysine is the common amino acid, and the atoms in black are what would make up lysine. And then pyrrolysine has this additional group attached onto it, which makes it pyrrolysine. So it has everything else that lysine would, but then it has this extra little group, which makes it different from lysine. And so these can get these can get incorporated into proteins dependent on the cell or the organism that may provide or help with the function of that particular protein. But they're not common amino acids and not commonly found. And then the last little part here, so we talked about proteins here. So if you consider all the proteins in an organism, in a cell, in a tissue, or just all together, this is referred to as a proteome. So it's considering all the proteins produced in a cell. So if you looked at just a cell and looked at all the proteins that it produced, that would be the proteome of the cell. If you looked at a tissue and considered all the proteins that that tissue would make, that would be the proteome of that tissue. And then if you looked at the whole organism and you considered all the proteins that would be made in all of the different cells within that organism, that would be the proteome of that organism. So a proteome is variable, remember, as that different cells will make different proteins. This is part of cell differentiation. So like a red blood cell will make different proteins compared to a muscle cell. So, a red blood cell will have hemoglobin in it, whereas muscle cells will not have hemoglobin. Muscle cells will have high levels of actin in it, whereas red blood cells will not have actin within them. So different proteins are made by different cells. A protein is variable, as different cells will make different proteins, and this is dependent on the genome, so it's what proteins the organism can make, and environmental factors. Temperature and nutrition can affect what proteins get produced. A cell can make different proteins over time, depending on the activities of the cell, depending on what stage of life or differentiation that cell is in. And then proteomes are unique due to small differences in the sequence of amino acids. So my proteome would be different from another person's proteome because there would be small differences in the sequences of amino acids that I make compared to another person. Even though those proteins would be the same, they would function the same, there'd be these small little differences that ultimately would not affect function, but would make the proteome unique to each individual person. So every individual, even identical twins, has a unique proteome as a result. So that ends this video for subtopic 2.4, protein team.